This is a huge area, and I would really like to cover it in as much detail as we can. We're going to go to our break after the next presentation, which is going to be given by Ed Bayer, and he's from the Department of Biological Chemistry at Weizmann Institute of Sciences in Israel, and he's going to talk to us about the potential for designer cellulosomes for biomass conversion out of the box.
Okay, well, I'd like to join my fellow speakers who preceded me in thanking the Baker Institute and the organizers for this kind invitation and opportunity to present some of our work. I have entitled my talk, The Potential of Designer Cellulosomes for Biomass Conversion Outside the Box. Now that didn't work. What do I press here? Oh, it's down here. They were hiding it from me.
There you go. I will do that.
Okay. Okay, outside the box, I actually took from our letter of invitation, in which I was asked to try to look outside the box to find non-traditional solutions to our ongoing energy predicament. I guess many of us go through life feeling outside the box. But anyway, in order to describe to you what we are trying to do, I have prepared a very extensive introduction, which starts, like our convener told us, in the beginning. For in the beginning, God, as we have been taught, created the trees and the other plants. But unfortunately, these die and become environmental wastes. But fortunately, we have the microbes which have evolved to take care of the environmental waste and to provide the nutrients back to the trees and the other plants. Now, this cycle has continued for time immemorial until man came on the scene. And what man has done is he has created lumber and agriculture. And from lumber and agriculture, the wood industries and paper industries, food and clothing, which has filtered through our society to become municipal solid waste and industrial solid waste, which looks something like this. And even from this vantage point, we might even be able to see that much of this is cellulosic in content. But anyway, the wastes are hauled off to landfills, mainly. Some is recycled, some is incinerated, some perhaps composted, but most to landfills, which becomes environmental pollution. This is the landfill outside of Tel Aviv, and I took this picture in February 1992. And I'd like you to just concentrate on the landfill and see what has happened in 6 short years. The landfill has completely filled up, and now our trash in Tel Aviv is hauled off to the Negev, the desert. National Geographic, 1991, a professor of archaeology published this article on once and future landfills. This archaeologist had been studying the trash of the Mayan civilizations in Central America. And at one point he decided, why not look at our own trash? Now in this almost prophetic picture that his photographer took in the early 1990s, we see the Twin Towers in the background, kind of obscure in the background. In the foreground, we see the landfill, which almost in premonition became the final resting place of the Twin Towers 10 years later. But Dr. Rathje tells us what we might find if we dig deep down into these landfills using these classic archaeological instruments that are shown in the figure. You might, for example, unearth a 7-year-old head of lettuce. Now, lettuce in my refrigerator is known to have a half-life of about 2 weeks left to its own devices. and is totally unrecognizable. This one, after 7 years at least, is recognizable, if not inedible. Corn, you might find a 20-year-old head of corn, 7-year-old baguette, or a 17-year-old hot dog. The take-home message is that organic material in a landfill is very stable. And what about paper? What about cellulose? Well, this is a 12-year-old newspaper which is perfectly legible. In fact, this is the way that they use the newspapers, the dates on the newspapers, to date the other material in its environs, the other items in the landfill. And it turns out that newspapers are the single most common item in the landfill. About 18% of the volume of the landfill of newspapers, and 50% and more is cellulose in nature. Okay, so cellulose, as many of us know, is in the plant cell wall. It is these, it looks like these steel-like rods, and indeed the cellulose is gives the strength to the plant cell wall. And in between, it is enmeshed in a colloidal type matrix, which is known as the other stuff. And this consists of the hemicelluloses and the lignin, and the pectins, etc. Most of it is polysaccharide, and then the rest is perhaps lignin. Now, what's so special about cellulose? Cellulose is a very simple sugar. It's simply glucose, which this is not for the scientists, actually. It's for the policymakers and the economists who might not know what cellulose is. So the scientists can continue to sleep. It's simply glucose in long chains. I've shown here 3 chains that are in parallel. The fact that they are in beta linkages all throughout means that the unit of cellulose is cellobiose, hence the name. It's a very boring substrate, being only glucose. But when we put all the chains parallel, this is how it looks. And the interesting thing, and what gives it its strength, is all the hydrogen bonding network that occurs, and this is what the microbes are up against when it comes to degrading it. And it is, of course, the microbes that we're talking about. And how do they do it? The bacteria and fungi in nature that deal with the cellulose, that know how to deal with the cellulose, produce tremendous quantities of the enzymes which degrade the cellulases. And there are many different types of cellulases. 2 types I've shown in this picture. There's the endoglucanase. This is an X-ray structure of an endoglucanase and an X-ray structure of an exoglucanase. And the difference is in the active sites. The endoglucanase has a cleft-like active site, whereas the exoglucanase has a tunnel-shaped active site. And this dictates the way it interacts with a cellulose chain. The endoglucanase, if this arrow is a cellulose, it depicts a cellulose chain, the endoglucanase can act on the chain anywhere along the entire length of the chain and severs the chain at various places in a random cleavage. The exoglucanase, on the other hand, like a noodle fitting through the little tunnel, the cleavage occurs at the ends, and we have what's called processive cleavage because it continues to be hydrolyzed in a processive mode successively while it's caught in the active site. And the 2 types of enzymes help each other so that they act synergistically, meaning that the sum is greater than the total of the individual parts, so that the endoglucanase creates more ends for the exoglucanase, which then can act processively and cleave it very, very quickly. Okay, but that's not the whole story. Cellulases are multi-modular enzymes. The catalytic module I've just described in the previous slide, but also have other types of modules. They're very complicated enzymes, very sophisticated enzymes. Some of them have cellulose binding modules which target the catalytic module to the substrate, and others have a Doctrine module that we'll talk about later, and there are many other types of modules. So cellulases are not what the normal types of enzymes that others who aren't in this particular field are used to. They are much more complicated, much more sophisticated, not simple enzymes. Okay, so any given bacterium or fungus will produce what we call a cellulase system, and this is one from the bacterium Thermobifida fusca, which is very similar to the fungal type enzymes. It produces a limited number of enzymes, 6 in this case, each of which has a catalytic module, and they all have also a cellulose binding module. So what's the consequences? Well, if we have the enzymes produced by the bacterium, which are then secreted, they then interact with the substrate, if it's cellulose, and they bind very strongly to the cellulose, and then they act synergistically together. And this was the dogma before we began our work on the cellulosome more than 20 years ago. We discovered what we described eventually as a discrete cellulose binding multi-enzyme complex for degradation of plant cell wall polysaccharides. Okay, our initial observation was a simple one, that bacteria, in this case, the bacterium Clostridium thermocellum, the cells bound very, very strongly to the substrate, and here you see the cells bound to these chunks of microcrystalline cellulose. And we were actually not even looking for enzymes. We weren't looking for, so we didn't know what a cellulosome was. We were looking for a CBF, a cellulose binding factor. We wanted to know why these bacteria bound to the cells. To make a long story short, after several articles, we described this multi-enzyme complex, which had many, many enzymes all connected to a single entity. And the other surprise was it had cellulase activity. It was enzymatically active, but not all the subunits were enzymatically active. There was this one very, very large subunit that was not, and even at the time we considered that it had an organizational type capacity. These are the isolated cellulosome particles, and if you look closely, you can easily see the different subunits on the complex. Okay, after the advent of molecular biology, it enabled us to determine the amino acid sequence of the various components, and we described eventually what we called the scaffoldin subunit. Now, this subunit is also multimodular. It has a series of modules called cohesins, which are all the modules are separated by linker segments, very, very characteristic linkers. It ends with a dockerin module, which I'll tell you soon what that's all about, and it has its own cellulose binding module. The enzyme subunits, on the other hand, are different from the enzyme subunits from the free enzymes in that they have the catalytic modules like the free enzymes, but they all have a single dockerin module. This is all shown schematically. Of course, this was all determined biochemically and from molecular biological evidence. And what the consequences of this is that the scaffoldin subunit attracts and binds very, very strongly to all the enzymatic subunits through a dockerin, cohesin-dockerin interaction as shown here. This is what we call the cellulosome, and you can already see that it looks like a molecular Lego, and this is how we are actually looking at it, as a molecular Lego. Now, it also has this cellulose binding module, and this actually drags the entire scaffoldin subunit together with all its enzymes to the cellulose substrate for subsequent degradation. But this isn't the entire story. There also, in time, we determined that there are anchoring proteins. The anchoring proteins interact with the single dockerin It has a cohesin on the anchoring protein, so we have a second type of cohesin-Dockerin interaction in the cellulosome. The anchoring protein anchors the cellulosome to something. It anchors to the cell surface, and this is why the cell is bound to the cellulose substrate through the single cellulose binding module that we showed in the scaffoldin subunit. Drags the entire cell to the cellulose substrate. And now we look at the entire complex as the cellulosome. Now, the enzymes of Clostridium thermocellum are much more extensive. There are many different types. There are cellulases, hemicellulases, and various others. Here I'm showing 20. This is what we knew in about the year 2000, before the genome came out. Certainly you can't fit 20 enzymes all together in 9 cohesins, which means that the individual cellulosomes are very heterogeneous. Each enzyme has a catalytic module and a Dockerin. Today we know that there are over 70 different modules, proteins, which have Dockerins in this particular bacterium. But there are other bacteria that produce cellulosomes, and we've discovered that every bacterium has its own story to tell. This one, Acetobibrio celluliticus, has an anchoring scaffoldin with 3 cohesins. It has an intermediate scaffoldin, which has 4, and this can attach 8 different enzymes, so 8 times 4 times 3. This particular cellulosome can have 96 enzymes on its surface. A different bacterium, a different story, Bacteroides cellulosalvans, has an anchoring scaffoldin of 10 different cohesins, a very large one. It's only superseded by its other scaffoldin, which has 11. So 11 times 10, this can hold 110 different components onto the cell surface. Okay, so we're not alone in this anymore. The cellulosome is studied by many people all over the world. The ones in yellow are our collaborators. The one in blue today are our competitors. But we're just one big happy family. We have our own Gordon Conference. This is the conference in 2005. Next year is the 5th Gordon Conference on cellulases and cellulosomes. Which brings us to what we're here for today. From conception, we've had a gestation period of 9 months, and until the birth of the conference, today's conference. And what do we have to offer? Well, we've seen the different types of biomass. So you could take plant fiber of any type. You might grow transgenic poplar trees. You might take agricultural residues, or even the municipal solid waste, any cellulosic material. And after the pretreatments that we have heard about, you might take microbes which specialize in cellulose degradation, or their enzymes, or together to produce the soluble sugars, which yeast or some other organism can then convert to ethanol, and then fill your tank and transport the plant fiber back to the pretreatment facility. Well, the enzymes might be perhaps one of the very major bottlenecks of this process. And what we have developed and suggested already 12 years ago was to use what we call designer cellulosomes for exploitation and management of cellulosics. Now, what is a designer cellulosome? The idea is to take different enzymes from whatever source, and through molecular one of the possibilities, the more interesting possibilities, through molecular biology, means to attach Dockerins to these enzymes, single Dockerins, to prepare chimeric cohesins with cohesins that will match the Dockerins, and then with the cellulosomes use them to degrade the cellulosic material. Now this, of course, this idea is not a new idea. I got it from playing with my children. The idea here is to take the different cellulases and put them together, but as we were becoming more creative, we started to create our own types of cellulases. Well, children mature, so do ideas, and eventually we are able to actually do this. And is it necessary? Well, if we were to take the artificial cellulosomes by breaking apart the cellulosomes and then trying to put them back together, the problem is that there is no specificity within a given cellulosome, and we would simply get complicated mixtures of cellulosomes, heterogeneous mixtures according to random incorporation of the different enzymes. And we were, of course, out for controlled incorporation for various reasons, one of which is that we like to control. And what we did was to take a chimeric scaffoldin, in which we sewed together through molecular recombinant means. We sewed together the different cohesins. These are now color-coded so that each one has its own specificity. We've taken enzymes with doctrines of matching specificities that we have created in the lab, and now we can control perfectly and incorporate a given enzyme into the chimeric scaffoldin. Well, we went to, in order to see how this works and so on, we went to our local paper mill to choose the correct type of cellulose substrate. We were offered various possibilities. In the end, we chose Avicell, which is commercially available. And if we take two enzymes which have the divergent doctrines on them and put them together, we get a basal type of activity of degradation of cellulose to soluble sugars. Now, if we add a cellulose binding module with a single cohesin, we now recreate the free enzymes so that each enzyme is targeted alone to the cellulose, and see how that works. And we get an increase in the interaction, which shows how much is due to targeting. And if we take our chimeric scaffoldins, we get a further increase, which is attributed to the proximity of the enzymes on a given scaffoldin. Now, we've done this with many, many different pairs. We've done hundreds of different experiments with hundreds of different combinations, and this is a typical type of stimulation or enhanced synergistic interaction that we receive from simply putting two different enzymes together in the same cellulosome. Encouraged by this, we went to choose a more complicated substrate. We chose straw, and then for the lab, we were looking for different substrates that we might want to use. In the end, we used a commercially available hatch straw, and we now chose a cellulosome that has three different cohesins. The scaffoldin has three cohesins. We use, in this particular figure, I show a combination of two different enzymes, 48F and 9G, from a given bacterium, and then we added individually 6 more enzymes. We get a very basal level of cellulases, and when we add the scaffoldin, we get a further increase in every case of the interaction. When we add a xylanase, a hemicellulase, instead of a cellulase to this mixture, we got a tremendous increase in the amount of activity because it's now it's now degrading the hemicelluloses. Well, in our lab, we've prepared for the future numerous scaffoldins. We've prepared numerous enzymes containing numerous types of doctrines in order to meet the challenge of the future. We have these all in our lab ready to go. Now, the question is, can the cellulosome actually degrades large amounts of cellulose? How does the native cellulosome fare? Now, this in many cases depends on the type of cellulose that you give it to degrade. For example, if you give it bacterial cellulose, which is a fairly easy, there's some bacteria which can produce cellulose, and it's actually very high crystallinity, but looks like these little ribbons of material, and it's fairly easy to degrade, and the cellulosome does so very quickly. Within a day, 100% is gone. However, if we take Valonia cellulose, Valonia is an algae which grows in two places in the world. There's a small version grown in the Florida Keys, and there's a very large version, it's about that big, a single cell, which grows only in Tahiti. So the people who worked on this had to every year go to Tahiti to get their material. They had grants for this. But it turns out to be the most crystalline form. It's almost 100% crystalline cellulose. It is very tough to degrade, but the cellulosome, even though it takes 16 days, is able to do so. Nothing else comes close. Okay, but can it really do the job? So we took the Avicell, which is a paper, it's a plant-derived form of cellulose. We took 2% of cellulose, and within a couple days, we got 100% solubilization. Then we took a more concentrated suspension of cellulose, 20% cellulose. We only got 60% degradation under the same conditions, but if we add an extra batch of cellulosome, we were able to get almost 100%. It turned out to be a thick molasses-type syrup mainly of cellulobiose and cellulose, and glucose, sorry. And how does it fare against the fungal enzymes? Well, we were working with some of our colleagues in France, and they had a fungal enzyme that they received from Novozyme, and they asked us for some of our cellulosome material, and I'm very happy to say that I did not participate in this experiment, because the results that I'm going to show you are theirs, not mine. So Trichoderma ricci, which is the standard fungal cellulase system, is able to degrade a certain amount of the cellulose that was given to it, and the Cellulosome fared much, much better, up to 6 to 10 times better, and the material was degraded that they gave to it was degraded within 2 hours, almost 100%. Okay, well, what can we do with it? Well, one thing is to produce once we determine which enzymes are best, we get larger scaffoldins, chimeric scaffoldins. We can then perhaps in vitro assemble the different cellulosomes and then use it for direct degradation of cellulose. Another possibility would be to take all the genes and incorporate it into a surrogate host, such as Bacillus or Aspergillus, or a solventogenic bacterium such as Clostridium acidobutylicum, or even directly into yeast. Now, I emphasize that all of these avenues are currently being investigated, and there are publications on this, not most of them by other labs. And then if we feed directly cellulosic biomass, we can produce the free sugars, or butanol, acetone, or perhaps directly to ethanol. Well, I'd like to end by thanking the various granting agencies in Israel, in the European Union, and in the States who have contributed toward our work, and my collaborators and students and friends in Israel, in the United States, the United Kingdom, my friends from Marseille, Portugal, and Italy. And I'll just leave you with a few pictures. This is my student, Henriette, from Holland. I'm the one on the right. I'm putting the arrow so you might not get us all mixed up in the next several pictures. Here I am with Brian White and his student Dion Antonopoulos. Here's a close-up of the three of us. Here I am with Mike Himmel in Denver. I'm the one on the left, my friend Mike Himmel. Here I am with his group. This is Mike Himmel's group. I'm over there. Here's a close-up of us. And my own group picnic a couple of years ago. We have an annual picnic, and with that, I thank you all.
Thank you, Ed, for an inspiring presentation. I know there are questions. Yeah, start over there.
Yeah, I was happy to see that you pushed the Clostridium. It kind of makes it more apparent that butanol may be the fuel that we ought to be looking at. I've been struck this morning by the fact that ethanol seems to be the major component here that everybody's talking about, but has the decision been made a priori by this group that ethanol is the king of alcohols and that we're not going to go the butanol route?
I think if you look at the program, there's going to be a butanol lecture.
Right by George Bennett.
I don't see him here, but I hope he
He
arrives.
will show up. I hope.
Yes, I was wondering. Anyway, I don't know, does anybody want to contribute to that
I think
question? Because I think it's all up in the air, actually.
I think ethanol has been mentioned today and yesterday because it's primarily, well, it's a substrate that we're looking at right now, but certainly with the mixed alkyl system and other systems, as you get to higher alcohols, certainly you're looking at much higher energy proficiency. You can utilize infrastructure that already exists, and I can say, at least for Texas A&M, we're not necessarily tied down to ethanol. In fact, we think the higher alcohols are much more viable biofuels.
Yeah, and I could add for BP, we're doing considerable work with butanol because we think it presents a much better fuel molecule while achieving all the benefits of a renewable fuel, and we're looking at how do we economically make it a marketable product.
There's not just Clostridium acetobutylicum, there are other Clostridia, which I'm sure you realize.
There's a two-stage process that does it very efficiently. I'm just thinking in terms of the recommendation of this group to energy and policy makers, something's got to be said to the public about the ethanol versus butanol debate. Everybody seems to be putting the emphasis on ethanol, and it would seem that a group like this could come out and say once and for all, let's look at butanol.
Well, there will be a white paper prepared after the end of this conference. This was actually our intention from the very beginning, to bring together the experts who will speak about ethanol versus butanol versus methanol, and who will speak about the politics of biofuels. And there was an interesting discussion yesterday about what is actually going within the beltway of Washington, D.C. concerning this issue. So we hope to bring all these things out.
Again, I'd add, I don't think it has to be versus. I think that's shooting inside the tent. I think what you have to really look at is what are the attributes of each one and put them on the table. Ultimately, I think those decisions are made by the marketplace. So I think you have to look at it from the viewpoint of developing technologies and identifying options to make them more competitive. But I think it's really dangerous for people who are working in the same field to be shooting at each other.
Well, the versus wasn't meant in an antagonistic but more likely a synergistic sense, because I think several people pointed out yesterday that there's got to be a global optimization, so to speak, of all these options. Okay. So, any other questions before we go on to the break? Jim?
I just wanted to ask about the enzyme loadings. When you were comparing the cellulosome with the fungal systems, you didn't talk too much about that. Can you talk about comparison on a specific activity basis, or?
Yeah, it's very hard actually to determine specific activity, because then you talk about specific activity against what? I mean, if you're talking about, exoglucanases have very little specific activity on various substrates, but they're very important because they're important in the synergistic interaction with the endoglucanases. And every endoglucanase, its activity on the normal endoglucanase substrate, which is carboxymethyl cellulose, is very different than its action when put into a scenario with pure cellulose or plant cell wall. So it's very hard. So what we do make a kind of a specific activity, or not activity, but we use pound per pound or something of that nature, kilogram per kilogram, to determine how much, and not even enzyme per enzyme, because the cellulosome has non-enzymatic components. Not only that, the cellulosome, as I told you before, has tremendous percentages of cellulases inherent in its when you grow it on cellulose. I mean, it's all and they're all heterogeneous. So it's hard really to control that. Okay, but basically we just took a certain amount of the cellulosome versus a certain amount of the fungal enzymes that were partially purified in that case.
Okay.
I think there was another question. Yeah, Bill, go ahead.
It's obvious you've made a number of different cellulases with the binding proteins. Have these been done with so-called traditional methods, or have you and or your colleagues or your competitors looked at things like DNA shuffling, since you obviously have very nice insight on the structure, function, the active site, things like DNA shuffling and putting together a high throughput, you have the substrate, putting together a high throughput assay might lead to another level of, let's say, enzymatic activity.
Now we've touched on this, but we haven't gone to anything more systematic than that, okay? We have done gene shuffling experiments. We've done a lot of mutations on the given enzymes. In our lab, we've concentrated more on the cohesin-Dockerin interactions and the cellulose binding modules, because these are more specific for cellulosome, whereas in other labs, the enzymes have been more the focus, okay? Others have used the isolated materials directly from the cells, okay, that have been grown. Others, like us, have concentrated more on the recombinant enzymes and so on. And we can actually prepare recombinant enzymes which are at the same activity levels as the native enzymes, okay? But the comparison is slowly but surely becoming more global.
Well, thank you again, Ed, for the presentation. Let's take a short break and reconvene here for the continuation of our technical program after about 10 minutes, around 11:15.
I'm going to have it.
Okay, let me start doing my final checks.
Yeah. Watch. Yeah.
Hey, Richard.
Sorry, I missed your paper. So go.
I have to get the speakers back. Well, the next three talks of this morning session are going to be on biochemical conversion. And we're going to start with Jim McMillan, who is with Biorefining Process R&D, again from the National Renewable Energy Lab. And he's going to talk to us about the biochemical conversion of cellulosic feedstocks. Jim?
Thank you. Wow, it is really hard to see from up here. All you can see it, I assume. Yeah, I understand. So I'd also like to thank the the Baker Institute and the conference organizers, conveners for inviting me. It's a pleasure to be here to speak to you about biochemical conversion. Now, we have heard the ethanol butanol thing, and I'll just bring up the biochemical conversion of cellulosic feedstocks is key, whatever we want to make from them. We've got to break them down. And so there's some central elements here. My examples will be ethanol-centric, but we'll be hearing later about butanol. NROLs recently, we've been asked to include this disclaimer. Rich didn't leave it up as long, but just to tell you not to make financial decisions based on what I'm going to tell you today, or at least don't blame me. Don't blame NROL. I think that's what that's about. So the speakers earlier this morning have set me up really nicely, and I will just try to shed a little more light on the subject of biochemical conversion. We've heard about the biomass opportunity. Really, it's moving beyond grains to the cellulosic resource, this is the most abundant carbon material on the planet, renewable carbon, that is. And it's a key to really going big with biofuels and biochemicals, but fuels being the bigger market. If we converted all the corn in the country to ethanol, we'd be somewhere in the 20 billion gallons of ethanol range. We need 60, 80, we need 100 billion gallons. We need much larger amounts, and the only way we're going to get close to that is with cellulose. And we can get to very large levels on the 60 billion gallon level based on this USDA DOE billion-ton vision study, which concluded that if we get serious about it, we can sustainably collect off our lands about 1.3 billion tons of biomass annually. I want to emphasize that there's a lot of there's some conservative assumptions in there. That does not include municipal solid waste. It doesn't include animal waste. There's a lot more cellulose there. It also doesn't. It makes modest projections about the rate of increasing the yields of energy crops like switchgrass. This is not available today. I think yesterday somebody made it sound like we have this billion tons. We probably have several 100 million tons of feedstocks right now. But to go big, we have to get serious about collecting this material. And one of the biggest points I would make, which is a little outside of the conversion area that I'm going to talk about, but it's that infrastructure, and it's how we do the sustainable agriculture. And we heard Professor Powers and Alvarez yesterday talking about some of the environmental impacts, and this whole sustainable production of agronomic materials is a big issue, and it's something that the students and all of us are going to be grappling with. How can we do this sustainably? Because we don't want to be clear-cutting our farmland, so to speak, and ruining their productive potential. So that's a very important thing. So but anyway, going big, the 1.3 billion tons, the study's conclusion is that with this 1.3 billion tons that we can achieve, that would be enough to displace about 30% of our gasoline. So that's a very current gasoline use. That's a very significant amount. It will not completely eliminate our dependence on petroleum, but it could do a lot to mitigate our dependence on imported petroleum. So in the talk today, I'd just like to go into a little bit more detail on biochemical conversion, talking first about the composition of the feedstock. Let's get a little bit more into that. We've heard some of it, so I'll go kind of quick. The main routes for biochemical conversion, and then focusing on the enzymatic hydrolysis pathway, which really is the focus of most of the R&D going on these days, and talk about some of the remaining technical barriers. And I'll do a lot of my comparisons with corn, since corn is what we're using today. We're producing about 5 billion gallons of ethanol annually in the United States, just surpassing Brazil maybe this year. And that's all from corn grain. So that's the material over there on the left. It's nice, homogeneous, pretty dense material. And what we're talking about is moving beyond that to the lignocellulosics. An example would be the the stover material in the middle there. And what stover represents is the above ground residue after you harvest the grain. And equivalent would be like straws from wheat and rice. And here we are at Rice University. I keep seeing the name Rice and I think of biomass because that's my orientation. I'm sorry. But you could probably figure out a way to exploit that. But it's not just corn stover. Corn stover gets a lot of play because corn is the biggest single crop we have, but we are talking also about the woody side of the feedstock space, which would include short rotation hardwood like poplars and energy grasses like switchgrass, which is a native prairie grass. That's what our plains used to be covered with. And so these materials look pretty different. You've got a pretty homogeneous material in these, and especially in the case of stover, where you have stalks and you have the rind and the pith and all this different complexity, very different physically. But when we burrow down and we look at the chemical composition, these things are sugars. There's 2/3 to 3/4 sugar in this material. This table compares corn grain composition versus the 3 cellulosic feedstocks that I'm just showing as representatives here. And you can see, whereas the primary component in grain, of course, is starch, which is a glucose-based molecule, much like cellulose, different bonding structure, but it's still glucose-based. You have hemicellulose and cellulose being the big players in the cellulosic feedstocks, but there are also carbohydrates that can be broken down to sugars that can be fermented to various products. The other big component we've heard about is lignin. Lignin is pretty recalcitrant to biochemical conversion as we know it today. I think it's an opportunity for some future work, but it's an excellent substrate for thermochemical conversion, and one of the themes that the Department of Energy is really trying to work on here is the complementary joining of thermochemical and biochemical technologies to maximize the production of useful fuels and chemicals out of these materials and improve the value proposition. Another thing I'd bring up is there are some other materials like proteins, a fair amount in corn, but there's also, say for example in switchgrass, a fair amount. And I don't know of any crops right now that are raised for only one use, and so I would posit that the bioenergy crops of the future, we're probably going to figure out some other things to get out of those crops besides just fuels. Looking sort of at the molecular structure, we won't go into the cellulose in as much detail. That was nicely done by Professor Baer. But we have lignin, which is this aromatic-based molecule, very energy rich, and as I said, pretty recalcitrant to biochemical hydrolysis. We have hemicellulose, which is if you will, the second most abundant, or the second biggest carbohydrate component in biomass, with xylose and xylan being the single largest. Xylan is the polymer, xylose is the sugar that makes it up, being the largest constituents in most of the types of cellulosic materials. Well, all the ones I showed you today. The only types of biomass where xylose is not the big hemicellulose polymer, where xylan is not the polymer, is softwoods, and then you have glucomannans, and it's more of a hexose-based material. But the xylan and xylose and its cousin arabinose are 5-carbon sugars, and this does present one of the challenges to cellulose conversion, and I'll talk about that a little bit more. Cellulose, we already heard from Ed, all the hydrogen bonding that goes on. I like to talk about it as molecular Velcro, and we got to break that down. And if we try to do that thermochemically, if we try to just apply chemistry like acids or heat, we'll break down a fair amount of that cellulose in getting to all of it. And so this is where we can exploit the selectivity of enzymes to convert cellulose at high yield without getting all the degradation. And this is, biotechnology in general has a big role to play all the way across the value chain, if you will, the supply chain in the feedstocks, in the depolymerizing enzymes, and in the microorganisms. And that's where there's a lot of energy behind this route, because we believe we can go a lot further with it. And that sort of takes me to how are we going to convert these materials. And I talked about a combination of biochemical and thermochemical approaches. The Department of Energy is focusing on biochemically going to ethanol from the sugar, the carbohydrate portion of the biomass, and then thermochemically taking everything that you can't get biochemically and converting it thermochemically to mixed alcohols, trying to maximize ethanol production. So as Rich outlined, maximizing ethanol production out of the biorefinery. Now, you could substitute other chemicals there. This is what the current focus is and what we're funded to work on. This new paradigm, this conflicting jurisdictions, or we heard that yesterday. that biomass is moving from food feed fiber to food feed fiber and fuels. And this is a paradigm shift with agriculture, use and energy were kind of separate, and now they're coming together. And there's a lot of environmental implications. And so this is the thrust of the Department of Energy's program, but it's also, I think, a challenge for our society, and this for the policymakers, and the fact that we have, we have the Environmental Protection Agency, we have the Department of Energy and the USDA, all having very sort of siloed roles, and we need to continue to build and figure out ways to make that structure work for this new paradigm that we're trying to move to. There's several different pathways for converting, biochemically converting material. They can kind of be broken down into the sort of hydrolysis sugar fermentation route, which is a very conventional route. This is sort of how we do brewing, where we categorize them based on the method for breaking down the cellulose. So historical processes like dilute acid or concentrated acid have been around for 50 or over 100 years. And they have been used quite successfully to depolymerize, not quite as high yields as you can probably get from the enzymatic processes, but they're fairly mature. And then there's the enzymatic process. Now, the enzymatic process alone, as Charlie Weinman spoke about earlier, the enzymes alone are not sufficient to break down the biomass. If you just take raw biomass and add enzymes to it, you will not get very much depolymerization. You need to activate. You need to make that cellulose and the hemicellulose accessible to the action of the enzymes and susceptible to it. So typically a pretreatment is used, and I can mention a few chemistries, but certainly acid and alkaline and organic solvent-based and combinations of those are all things that are being developed, and actually the CAFE group is one of the groups that's looking at a wide variety of those. Another approach is gasification, and then converting the synthesis gas using biology. So this isn't breaking down the cellulose biochemically, it's actually using gasification to make CO and hydrogen, and then using fermentation organisms, a rather different class of them, to ferment that, to convert the CO and hydrogen into valuable fuel products. That is I would say it's an area of an active endeavor by a couple companies. It's a little further out there in my opinion, and it's not the focus of the rest of my talk, so I'm not going to talk a lot more about it, but we can go there in the questions if you want. Why are we emphasizing the enzymatic route? Primarily because the upside potential for getting to low costs and very nice economics are the highest there. There's, as I mentioned, the concentrated acid and dilute acid processes have been around for many, many decades, and we're fairly far along on the learning curve. So how much better are we going to get with those processes? Probably not a whole lot better. Whereas with biochemical conversion, biotechnology, there's lots of room for improving the specific activities of the enzymes, for improving the feedstocks themselves, and improving the fermentative microorganisms. And we've only begun, and the tools are just getting stronger and stronger, and our belief in what we can do there getting stronger. So the upside potential for low-cost production is best. So that's really the why. So now I'm going to go into my let's talk about the process, and I want to compare it to starch, to corn grain conversion, because that's what we're making the 5 billion gallons of ethanol that we're making annually. We're making it from starch. And so what happens? It's a pretty simple process at the heart of it, where you take your grain in, you mill it up, and then you mash it. Okay, that would be the brewer's term. And this is using a combination of acid, heat, and enzymes to liquefy, solubilize that starch, break it down to simple fermentable glucose sugar. Add a fermentative microorganism, typically Saccharomyces cerevisiae, brewer's yeast or baker's yeast, and carry out glucose fermentation. It's intentionally or unintentionally, we've been doing this for thousands of years. And we end up with an ethanol solution that we can distill off, and an unconverted solids portion that's valuable as an animal feed. That's where most of our protein is. And so we have a fuel and feed process. This same general concept applies also for cellulosic feedstocks, but because of the recalcitrance of the plant in the cellulose and the intermeshing of lignin and hemicellulose and so forth, we have to go to much more severe, if you will, cooking conditions or mashing conditions. And we really break that out now into 2 steps, which are pretreatment and enzymatic hydrolysis as separate steps. The pretreatment, as I said, in this case could be acid, alkaline, or an organosolve type process. Typically it would be carried out at 160 to 200 degrees C for a minute or so, a couple minutes. And it would be at some extreme of pH, low or high or organic solvents. So there'd be some, if you just threw enzymes in that solution, they'd probably inactivate pretty quickly. So you typically are conditioning that material, you're bringing the pH down to a range, physiological range. You're bringing the temperature down to compatible with the enzymes, carrying out enzymatic cellulose hydrolysis then on the pre-treated soluble, on the insoluble fraction. So in the thermochemical pretreatment, if you say used solute acid, you would solubilize the hemicellulose. You'd end up with a xylose-rich liquor solution and a enzymatically reactive lignocellulosic solid. That's what you would add your cellulase and or hemicellulose enzymes to. They would proceed to processively degrade that material down to glucose and perhaps the sugars from hemicellulose, which would then be, can then be fermented. Now, in this case, Saccharomyces cerevisiae doesn't have a substrate range that allows it to ferment many of these sugars, native strains, and so you have to use some sort of different fermentative microorganism, but there are both wild type and increasingly a number of recombinant microorganisms that have been developed that are quite effective at converting most of these sugars or all of them to ethanol at high yields. There are still some challenges there, and I'll get to that, but here the lignin has passaged through the process relatively unchanged. It is changed a little bit, but it's still there, and we can use that as a solid fuel to provide the heat and power to drive the process. And as Professor Wyman alluded to, there's a little bit of extra, on most of our calculations, there's extra lignin, if you will, and so we can actually, if we want to invest in a turbo generator and create electricity, we can also make an electricity co-product and sell that back to the grid. So we have sort of a fuel and energy process. Now, just you know, my theme would be, and we've heard, this technology is there. You can do this. This all works. And the challenge is making it economical. That's really the, you know, what we're working on. And this is some raw stover. This is the solid. If I just showed the liquids, it'd be, you know, brown water, brown water, brown water, so it wouldn't show so much. But here we have a corn stover that's been untreated. Here is after pretreatment, so we've extracted the hemicellulose. This was a dilute acid pretreatment. And here's after we've done enzymatic hydrolysis on the pretreated solids, and that's the lignin-rich residue. So you can do all this. And as I guess I'm going to move into the economics a little bit. We've done a lot of economic analysis at NREL. And this just shows a simplified view of what's considered when we do one of these economic studies. And so we bring the stover in. There's a section for bringing it in. You've got a solid material. There's a fair amount of work there. A pretreatment section, the sort of conditioning section, enzymatic hydrolysis and fermentation typically combined. As the sugars accumulate, they will inhibit the action of the enzyme, so people often combine in a simultaneous saccharification and fermentation scheme to increase the overall rate and yields of conversion. The ethanol that's produced is recovered by distillation, wastewater treatment, and then your lignin is combusted and perhaps converted to electricity in a boiler turbo generator system. And although this looks like it's just a little piece of the puzzle, it's about 1/3 of the capital cost of the plant. And so this is where the scales of economy issues come in, and we've heard that a few times, but it can't be neglected. Now, I said corn is the big one, so we've got to compare things to corn. This is a mountain of corn, or a hill by Colorado standards, I guess, of corn. But corn is really where the action is today, and really from an investor perspective, how far can we go with corn, and where is cellulose, and At what point does it become attractive for the next investment dollar to go towards cellulose? In mentioning that, I would just also bring up that, you know, my conclusions on the economics are based on what it's going to take to get to 60 billion gallons. And to do that, we're going to have to access parts of the feedstock space where the feedstock's a little more expensive. There's a wide range in feedstock costs from captive residues that are at plants like corn fiber or bagasse to feedstocks like energy grasses that you've got to go out and plant and harvest all yourself and pay for everything. You can't piggyback onto any other production process or crop value. So the economics that I want to talk about are getting to, you know, head-to-head with corn on a fairly expensive feedstock so that you can, so that the investment decisions will be to go forward and commercialize. This shows relative production cost for two conceptual processes, sort of that I outlined in that cartoon before, a conceptual dry mill and a conceptual enzymatic stover, corn stover process. And it just breaks out the cost. And what I've done is I've normalized both the cost to one. Okay, so you can just see when the technology is good enough, this is approximately probably how the costs are going to allocate, approximately. And what I bring your attention to is the importance of feedstock cost in corn ethanol production relative to cellulose, that the cellulose is less expensive feedstock. It represents a smaller percentage of the total cost of production. But what I would also bring your attention to is I guess first the co-products, that there's a very significant co-product in the existing corn ethanol industry. And the electricity co-product we envision is pretty modest. It doesn't play a big role, but there's a big cost associated with it. And then the third thing would be the capital cost. The corn dry mills historically have used natural gas boilers. They're bringing in fossil fuels for their heat and power. They have a much cheaper heat and power system. They're pretty, a couple decades of development, they've gotten pretty lean technology. So their capital costs are pretty low. We have the additions of a new technology, but a more recalcitrant substrate. And so we've got more pressure, longer residence times, more tanks, et cetera, lower concentrations. And as a consequence, we have a lot more equipment. And so that capital charge is very high. And we've heard that the capital is challenging, and it is challenging. It is the big challenge. So if you look ahead, you say the opportunities are really lower operating costs, potentially significantly lower. Of course, that very much depends on your feedstock, and there's a wide range of feedstock costs. There's also the opportunities to hedge against utility prices and corn price fluctuations, which we've seen in the past, and actually I've seen on the Chicago board futures, corn bounces up to $3 occasionally and stuff like that. If corn prices go up high enough, that will just be very damaging to the industry. And so Cellulosics would provide a way of bridging without incurring that or de-risking that. The capital cost is estimated to be in the $2.50 to $4 per annual gallon for Cellulosics versus only about $1 to $1.50 today. One thing that's interesting is, as Rich Bain was talking about, is a lot of the corn millers are now looking at putting in biomass-based heat and power systems. So their capital costs are actually going up as they're putting in solid boilers that can handle solids, basically, while progress continues to improve the technology for cellulose, and so the gap is narrowing. The other opportunity really is other products, and we're going to, I think, hear more about that, but there is a big challenge here. I mean, we've got a new process stream. There are new chemicals, and there's intermediate streams that we don't see out in current industry today. And so there is an opportunity for new molecules like xylose or something to become big chemicals in our industry. However, the scales of fuels versus chemicals, and especially transportation fuels, it's a couple of orders of magnitude. It's very, very large. So it's hard to, while you can see lots of valuable chemicals in there, it's hard to get them to be produced at scales where it's economical. So there has been a lot of progress. I'm going to talk about that in just a minute, but this is estimated minimum ethanol selling prices from the NREL model of the Stover process. versus time. This area over here is sort of our state of technology estimates, where we've taken bench and pilot scale data, the best data we have, combined with assumptions and stuff we haven't figured out yet, and projected what that cost is. And you can see it's dramatically dropped over the last 5 or so years to now being somewhere, and this differs from what Charlie's saying, but estimating around 220, 225 per annual gallon. Now, I just want to emphasize that feedstock issue. This is assuming $53 a ton feedstock. If we go to much lower cost feedstocks, that price would go down dramatically. The Advanced Energy Initiative's goal is to get that cost down to $1.07 by 2012. That was originally a 2020 target, and it's been moved up with that initiative to accelerate R&D. You can see that most of the cost reduction has occurred in the enzyme area. And we heard yesterday that enzymes were the bottleneck. They're not the only bottleneck, but they were a big one. A lot of progress has been made in bringing down the cost of those enzymes, but more progress is possible and would be beneficial. I'll talk about that. So sort of the 3 areas as I group them that we need work in are feedstock valuation and delivery, the biomass recalcitrance to conversion, and sort of the overall integration of the technology. The valuation area I'm not going to talk a whole lot about, but if you go out to that pile of bagasse or rice straw, how do you value that? What's the ethanol potential? What's it worth? What are you going to pay for it? We need ways of quickly doing that. We don't have a lot of those. And then the sustainable supply systems is something I already mentioned. That's just a very important thing that we do this right. The biomass recalcitrance to conversion is really the area that I work in, and the focus is on the key depolymerizing steps of pretreatment and enzymatic hydrolysis, and then on the fermentation of the sugars that you produce there to ethanol. And the big challenge is the pentose or the mixed sugars, the arabinose, the xylulose, the non-hexose, the non-6-carbon sugars that are present at substantial levels. And then biorefinery integration, you're really looking at the chemistry, you're looking at process interactions, there's a lot of them, and some of the solids handling issues that you get into when you're dealing with an insoluble substrate like cellulose. So that's what I wanted to talk about in the last few slides here. This is a picture, I think Ed Bear also showed a picture that was pretty similar to this. It's a representation of a primary plant cell wall. This does not show, so you have these large tubes are the cellulose microfibrils, the cartoon of them. The interconnecting blue is the hemicellulose, but there really isn't any lignin shown in here. I think we might have some pectin or some other things over here, but it's a primary cell wall, and where this mass of cellulose resides is in the secondary cell wall, where you have a lot of lignin. So it's a very complex network, and each of these polymers has different susceptibilities to chemicals, temperatures, whatever, and extracting them at high yields is non-trivial. And unlike starch, these are not storage carbohydrates. These are not made as an energy source that the plant can tap into. They're made as structural components. They're the bones, if you will, of the plants to keep the plants from blowing over in the wind, being attacked by insects, and so forth. So there are some reasons that it's harder to break down. So the cell wall recalcitrance is a big one. One of the things we've been doing to tackle, to get at that, we've got to operate at high solids concentrations. We've got to try to do things to make it more economical to run these pressure reactors. And so over the last several years, we have succeeded in getting pretreatments. This is the first step, and this is dilute acid pretreatment example I'm showing, but getting our continuous reactors to be running at 25 to 35% solids pretty routinely, which is quite non-trivial. And I have a picture at the end that will show you what this material is that I'm talking about. But so we have been bringing down, this is again the minimum ethanol selling price as a function of solids concentration to the reactor, and it is important to maximize that to bring down the costs. Enzyme costs have fallen sharply. This is just showing some work that Genencor and Novozymes did under subcontract to the Department of Energy. NREL played a validation role and enabler role in this. Both of the companies focused on proprietary approaches, but it basically reducing production cost and increasing the efficacy of the enzyme preparation so you could use less. And they went from several dollars a gallon estimated cost of the enzymes to under a quarter. And it just shows the power of biotechnology in action. And as Ed said, these are non-trivial enzyme systems, and there's a lot of synergy, and it's not just like getting an improvement in one component. You've got to do a whole bunch of things to make this stuff work. There was an award for this, some of this work 2 years ago. This just shows sort of the inverse Moore's Law approach. These were multi-million-dollar subcontracts, and you can see the cellulase cost per gallon as a function of time and the estimated cost, and it really did, I'm not showing the actual data points, this is a regression to them, but really the actual data fit this kind of line, and it shows that if we get serious about this, and this would be for the public policy makers, I mean, if we apply money to this, we can make progress, and this is progress. This just blows up the last 18 months, and you can see we're below that 20 cents a gallon by the subcontract metric. I would just emphasize at this point, okay, this is great. Enzymes sound cheap, but the metric used a wash solid, there wasn't feedback sugar inhibition, it was at a modest solids loading. So it's not maybe the most industrial relevant, it's pretty industrial relevant. We tried to make it mimic as much as we could the base case economic design that the numbers I've shown you earlier were from. But we're still using on the order of 10 times more enzyme protein for an enzymatic process than we are for a starch process. So there's definitely room for specific activity improvement here, in my opinion. Or for other ways like enzyme recycle and things like that that would allow you to greatly reduce your net use of enzyme. An area that we're going into now at NREL that's pretty interesting and really exciting, I think a lot of the folks that work on the process side like myself are having fun with these tools and getting to play with them a little bit, is starting to look at things at the, if you will, the ultra structure of the cell wall. You know, I talked about hemicellulose, cellulose, lignin, you know, like there's just one level of it, but where is it? How is it organized? What is the structure? When an enzyme is actually sitting there, you know, it's on the order of tens of nanometers and it's sitting there on a site, what does it look like? And so we actually, I think it's our society, you know, the science has come along so that electron microscopes that used to be, you know, the size of a building or can sit on your table, on your desk. And so we can start doing this kind of work. And this is just an example. It's a cross-section of a corn stem and some various blow-ups of it. But so we're able to look down at the nanometer scales and start understanding how enzymes and chemicals are penetrating these materials. And now think if you can genetically engineer the plant and start using these types of tools and getting that synergy going and figure out, design the feedstock with the end in mind. There's some really interesting opportunities there, and also just to understand bottlenecks in the technology as we know it today. Another barrier is pentasand utilization. This is the C5, so-called C5s. This is a carbohydrate fraction of these different feedstocks, and you can see, you know, starch, it's just glucose. We've got very significant fraction of pentasands, primarily xylan, but also some arabinan. In all the cellulosic materials. And this presents challenges because these pentasands don't come in through glycolysis. They aren't metabolized the same way in cells. This just shows a putative metabolic pathway for xylose fermentation in Zymomonas mobilis, which is an organism that's been engineered at NREL to expand its substrate utilization range to include xylose. And this is the regular glycolysis pathway. It's actually the Entner-Duderoff pathway. It's a slight variation of traditional glycolysis by EMP pathway, but glucose comes in by this pathway. Xylose is believed to come in through the pentose phosphate system, and you have to introduce enzymes, because it doesn't have native ability to use that, to incorporate xylose. Then you need to muck around with the pentose phosphate cycle enzymes to make sure there's enough activity that you can shuffle, if you will, the pentose 5-phosphates into things like fructose 6-phosphate and glyceraldehyde 3-phosphate, which can feed into your standard glycolytic pathway. I'm not going to go deep on this, but just the fact that there's some inefficiencies maybe in the fact that it needs more enzymes, you don't have immediate capture of the sugar. We don't quite understand the reason, but these types of organisms, and there's now about half a dozen of them that are quite good at fermenting pentoses, they still don't have the hardiness and the robustness that we're used to and that industry is clamoring for that we get with yeast, brewer's yeast, where we can make 15% alcohol, where we can withstand contaminations. We have lots of, many decades of experience running very large fermentation systems. So that's the challenge, I think, is getting to that next level of robustness. These organisms are typically producing 5 to 7% ethanol, and at half to a third the rate of a typical glucose fermentation. The third area is process integration. This is really where I spend most of my time, and Charlie already talked about this, so I won't belabor the fact, but how you put these different pieces together makes a big difference, and especially like the pre-treatment and the feedstock also. You vary the feedstock, you vary the conditions for the pre-treatment, you vary degradation, you vary the sugars you get, the native digestibility and so forth. So this is an area that requires, and there's a lot of different feedstocks, a lot of different pre-treatments, a lot of different enzymes, and so it's also just sort of bewildering in the number of options. This is an example, though, that we can integrate these technologies. This is a 10 to 20 liter scale, simultaneous saccharification and co-fermentation. We're fermenting glucose and xylose together. This is concentration or total CO2 versus time, and just showing, so we've got the pre-treatment liquor, the hydrolysate is rich in xylose, so here's the xylose being fermented away. At the same time, the enzymes have been added and they're producing glucose, and the glucose and cellulobiose are again being fermented away. The cellulobiose is being converted to glucose, which is being fermented. At 96 hours, and then correspondingly you're getting ethanol production, CO2 production. At 96 hours you've consumed all your sugars. It looks like. You continue to make ethanol, and that's because you continue to hydrolyze cellulose, but as the glucose is formed, it's fermented away to ethanol. In this case, as I was alluding to, we make about 5% ethanol, and that was pretty good when this was done. It was a couple of years ago. But the point is we can integrate these technologies. But we're only getting to about 5% sugar. So our challenge is what we're working on at NREL, my group, is to try to get the concentrations up. And so this is just some data showing initial insoluble solids levels versus the sugar concentrations that we can produce. And you can see we're getting up into the 175, almost 200 grams per liter. So I've just got one last slide. Actually, I might have two, but let me see. Sorry. This is, I just wanted to, so I said that, I don't know if people are going to be able to see this. Is there any way to dim the lights at all? Or, but this is pre-treated corn stover at 45 degrees with an enzyme loading of 20 mg per gram. The reactor is actually insulated, and we're just opening it up periodically and taking a picture. So this is kind of a compressed time-lapse sort of, time-lapse thing. And I don't know if you can see it, but it's very solid. There's no movement at all of this material. You start to see some wettening, some glean on the surface by 4 hours, and the enzymes have been impregnated in this material. But by 8 hours now, you're starting to see some plasticity in the material. By 12 hours, which is coming up here, it is starting to flow kind of like a mud. This is, as I said, this is this is 25% insoluble solid. So when we're running our pre-treatment reactor at 25 to 30, you can see how concentrated that would be. But this is the kind of solids concentrations we're going to need to be at to be able to get competitive head-to-head with the current ethanol industry. So I won't belabor it, but you can see the stuff is pooling, and we could pump it over to a stirred tank reactor at this point if we wanted to. So I will just wrap up by saying that there's a lot of progress being made. The compelling operating costs are within reach, I agree there, especially for the lower cost feedstocks. That the high solids processing of the type I just showed is helping to reduce the capital, and industry is very much investing in this technology. in this area, and as a result, the commercialization risks are being reduced. There's more needed. I've already talked about it, but we got to prove the processes at scale and prove their benefits. And breakthroughs that some people in this room could contribute to could help to accelerate the rate of progress or deployment of this. And the recalcitrants, the ethnologens, new products are all areas where we could get a lot of benefit that could really spur things forward. So thank you very much.
Thank you, Jim, for a very nice presentation. Are there any questions? Tad.
I just have one brief question about scale-up. At the lab scale, you probably can disinfect your biomass prior to processing it very nicely. But the industrial biomass will contain fungi and bacteria that come with it. And when you then process it for 70, 90, 120 hours, these bacteria will start doing their job, converting, I guess, ultimately things to methane or some other products. Can you tell us a little bit about the competing processes to the ones that you described?
I can tell you that under extended integration, we have seen contamination issues, but generally, contamination has not been a big issue, and we're not doing anything to sterilize the biomass. I mean, we bring in biomass, we get it by the ton, without any, sometimes it's actually molding, or it's like an industrial captive material that's wet, so it starts to spoil. So it definitely has a very high biological load coming in. But we go through a pre-treatment process, and that pre-treatment is essentially, inactivating a lot of that material. Also, configurations like the simultaneous saccharification and fermentation scheme, the sugars laid on, there's no sugar there. So there's nothing for a competing organism to really get a foothold in. So I do think it is a scale-up issue that people need to look at. It's very dependent on the feedstock, the process, the microorganism you're using, and a bunch of things, but I don't see it as a showstopper. Right now, in advances in the dry milling industry, they're going to eliminating the cooking. So they're just using different, one of the variations is just to mill finer and add more enzymes. So again, validating the SSF kind of principle that if there's not a lot of free sugars there, that contamination's not a big deal.
In the interest of time, I think we should move on. There is an open time at between 2 and 3, so I hope that the morning speakers will be available so we can start, so we can have the opportunity, I should say, to ask them more questions, and in general have a nice discussion about the overall processes we're talking here. The next two presentations are going to be from Rice University. The first one given by Ramon Gonzalez from Chemical and Biomolecular Engineering on emerging platforms for biofuels and biochemicals, the role of metabolic engineering in systems biology. We see that we're now expanding on some of the issues that have been raised by earlier speakers. Ramon?
Thank you. Thank you for staying around, and we're going to try to make it not very painful and long so that we can all go for lunch. I think that, is it that I don't see it well from here, I guess, right? But you'll be able to see it? I think that he's projecting there
Very nice presentation.
everything. So I don't know if they... Okay, let me try here.
5.
Okay, good. Thank you. As Dr. Sigurakis mentioned, I will be talking about the title is Emerging Platform for Biofuels and Biochemicals, and specifically I will try to address the role of metabolic engineering and systems biology as enabling tools for achieving those goals. The previous speakers, I guess that will save me a lot of time, a lot of slides that I had for introduction that you have heard already. So you will see from time to time that I'm going to skip a slide is because of that. But first, let me tell you a little bit about what metabolic engineering is and what systems biology is. I'm going here, in fact, by the original definition given by who is considered the father of metabolic engineering back in '91, and in fact, Rice alumnus, in fact, a graduate PhD from our department, James Bailey. And the definition is, I guess, pretty straightforward. It's improvement of cellular activities, and this can go from you're thinking about plant feedstock to microbial system that would be used for fermentations. So in general, improvement of the cellular activities by manipulating enzymatic, transport, and regulatory functions. The key thing, of course, is the use of recombinant DNA technology. And the type of metabolic engineering cycle that most of us use nowadays is the one that is depicted there to the right, that involves a lot of omics type of tools, those that we are borrowing or that were made available after the sequence of the genome of many organisms. In the case of system biology, it's a little bit loose, the definition. It depends on who you ask to. So just opted for putting a very general definition there. It's a relatively new field that aims at system level understanding of biological processes. What is not loose is definitely the two determinants of system biology, although depending on the person you're asking about the person might emphasize number one or number two, but definitely system biology is both. Is basically it's embracing technologies for high throughput, quantitative, system level characterization of cellular functions, and also is needing a lot of tools in the area of modeling, simulations, and as described in the slide, advances in all those areas that would allow you to do at least a couple of things. I didn't want to take it too far, but at least data analysis, the analysis of the data that you are generating with the functional genomics tools that I'm talking about in the part number one. The integration of that data, that's very challenging given the amount of data, the high dimensionality of the data produced by those functional genomic tools. And of course, we never quit trying to predict things, so although I didn't put it in there. This slide, you had seen variations of this one, trying to make the same point. We're trying to establish a parallel with the petrochemical industry, and in such a way we start with similar, there are different feedstocks depending on which part of the diagram you look at, but basically there are very lot of similarity between the petrochemical and the bio-based industry, and it has been discussed which are the advantages and so on, so I'm going to skip that. I'm going to make sure that you know that the main three platforms that we have now, and I might be missing a little bit here, but the main three that you have is the sugar platform, and is defined by the fact that that's the part of the feedstock that we're using, the plant biomass sugars, the syngas platform, and then the oil platform, and the speakers today have talked about all these. Again, the oil platform, you're using the oil as feedstock, and the most well-known product out of it maybe is biodiesel, or at least the one that is more in the news. I will talk about a couple of them only. The sugar platform, I'm focusing in lignocellulosic, and then the oil platform, but not exactly about biodiesel. I will tell you in a few minutes. Emphasize the point again that the future feedstock, the one for tomorrow, the one that's going to have an impact is lignocellulosic biomass, and that's what we're talking about, emerging technologies rather than the corn. And as was pointed out before, there's several challenges, and the one that is closer to the type of research that I do is the challenge of fermenting sugar mixtures that was pointed out in previous presentation as well. This is the diagram, but again summarizes the whole process going from cellulosic biomass to whatever product. In this case, it's ethanol, but I put there ethanol, butanol, your molecule of choice, although this is referring to ethanol here. One thing that I wanted to point out was Jim in the previous presentation talked about this part here, that is simultaneous saccharification and fermentation and different versions of it. There are people also, and this is I would say very emerging in terms of not being established at all, is the one that is represented by the whole blue rectangle there, that is consolidated bioprocessing, which you will try to consolidate the whole thing in one box. But other than that, what is trying to be, what I'm trying to illustrate here has been illustrated before, so I'm going to move ahead, and this is a different version of the different parts that this problem has, or that this process, So you want to produce fuels and chemicals, you have to get the appropriate feedstock, and we had talked today about it, feedstock deconstruction, and we had talked about it as well. The part that I work, that my research is focusing on is the microbial fermentation. So biocatalysis, you can have different type of transformations there, but the one we focus on is within biocatalysis is microbial fermentation. I'm going to focus my talk on the application of microbial fermentation in a couple of these emerging platforms. If you think about microorganisms that would do the job of fermenting the sugars and getting all these kind of things that you have in here, this is a list of traits. I typically call it a list of desires rather than actual traits. I don't know if we will get down to achieve all that, but definitely it's not, in fact, it's not complete the list. And then you have choice of different organisms, and from there, of course, again, is incomplete the list. It's just giving some examples. And the ones that are highlighted in a different color, that by now I don't know what the color is exactly, but different color are the ones that I'm going to talk about today. So I'm going to talk about this organism, and that is a bacteria, E. coli, a benign E. coli, not one that is going to have you with problem with food poisoning or anything. And then I'm going to give certain examples to illustrate the application of metabolic engineering and systems biology in this area. And they're going to be focused in two things. The substrate utilization, basically 5 and 6 carbon sugars, and the tolerance to ethanol. Again, the idea is for example, in the case of sugar utilization, is the simplest way to put it is you're going to have a mixture of sugars. All this is just an illustration, of course, so I am simplifying it by putting 2 sugars. And the simplest problem maybe you're going to have, and not maybe exactly the simplest one, but the most common, is that glucose is going to inhibit the metabolism of other carbon sources, these 5 carbon sugars, mainly xylose and arabinose, the most abundant ones. And you're going to have this sequential utilization of the sugars, and many times, depending on the concentration that you use, you might not even consume completely the other sugar. And well, what is it that we want in terms of the phenotype, that's the behavior of the organism? This is the type of things that we want. Now, what I'm going to do next is try to take you through the type of regulatory system that control the utilization of sugars briefly, and show you how, using a systemic or system-wide approach, we have identified or we have discovered certain characteristic of that system that would be useful in the future for engineering the desired strains, but as of now, they are providing knowledge about the system. And talking exactly about that, that's the philosophy. You understand it, so you can harness that later on. It's very, very popular nowadays, an approach in which is called evolutionary engineering, in which you maybe you claim that you don't need to understand it, but even if you want to use evolutionary engineering, you need to have a minimum understanding of the system. So, however, we do use mainly the type of approach that is based on the understanding of the system and introducing modifications based on that understanding. Of course, the system that regulates the utilization of sugar is relatively complex. In fact, in this diagram here, what we have is at the bottom of it, all this is about regulatory pathways. And the actual metabolism of the sugar is taking place up here. I am putting different colors, glucose, xylose. Again, I didn't put arabinose because we didn't have it in this illustration here. And you end up producing ethanol. Again, this is an example of a product. And the same would be true similar for butanol or any other product. So if we want to get real understanding of that, we are all advocating in this area, from, again, feedstock engineering to microbial fermentation. Anything related to biology is taking a systems biology approach, a systemic approach. And you're going to see that in a lot of reports out there from DOE and other sources, and most papers you read nowadays. And that is because of the availability of genome sequences for many organisms that let you develop tools that basically you would have an overview of the whole metabolism, an overview of the whole organism inventory of mRNA, proteins, etc. So we claim that we want to use that approach. And again, if we want to use the system biology approach to understand this, we need to have those two components that I mentioned at the beginning. One is more experimental, based on high throughput technologies, and the other one is more toward the modeling part. And then when you think about this, I typically use this diagram here, that is shown bigger in this slide. And the idea is the outer layer illustrates the relationship between genotype and phenotype. And for those of you not having background in the biological area, we're talking about genetic information and behavior. As in the case of humans, we behave in a given way due to two general things: our genetic information and the environment. And the environment includes everything else but the genetic information. Of course, that's very simple to say, but the point is, when we talk about metabolic engineering, what you want to do is to design a given genotype that gives you the phenotype that you want. So cells that consume a lot of sugars, that produce a lot of ethanol, and so on. If you want to do that in a rational way, you need to have some degree of understanding of this relationship. And that relationship is taking place through a lot of processes, and is oversimplified in this diagram, and is oversimplified with the objective of showing you for which of the areas in those processes there are available tools out there that allow you to survey the whole metabolism, meaning high throughput functional genomics tools. And that's the case for, again, surveying the expression of each and every single gene, for example, in E. coli or other bacteria and other organisms, surveying large fraction of the proteins that are expressed or not in given organism. Same thing for metabolites, and same thing for fluxes that is not actually a species, is an event, is the rate of the reactions that are taking place. So if we go back to our slide, and fortunately it worked, you're going to see that I put little pictures here illustrating basically the tools that are available for the different parts of that pie. And the one that I am missing is the one for genomics, and that's the sequence of the genome of a given organism. So how do we put all these together, right? We said we want to put it together. How do we do it? I have been thinking about this, and I thought the best way to illustrate it is using this cycle. And for those of you that have thought a little bit about the cycle of knowledge, this is basically the same thing. So if you're going to look at a system like this one that is let's say E. coli fermenting a sugar mixture, you're going to start with the given knowledge that exists already. Now, based on that model, you're going to establish a conceptual and or mathematical model. We all do modeling in our minds, even if we don't put it into equations. Using that model, we are going to have predictions, and definitely you're going to formulate hypotheses, and you go around this cycle by conducting experiments. And then in this portion here, what you have is inductive inference. So based on the results that you get of your experiments, you're going to infer something, then you have the synthesis step, and then you get updated knowledge. Basically, what we are doing is exactly that, developing tools for completing this type of cycle and using the cycle to understanding these systems. This one is small, but you already saw this. And the aim of this is not for you to actually see it, just remember that it is a given regulatory and metabolic network in there. So if we start, like in this case, the first thing that we need is modeling tools that allow us to predict things, and so that we can formulate hypotheses. We can do this by, bare hand. So we can think about the system and predict things without, systematizing anything, but of course that's not the optimal way. And in there we have developed a tool we call, is a kind of different type of approach, and we call it elementary network decomposition. I hope that the link is going to work here. Good. So the idea is that we actually break down the network. Given network is going to have a bunch of components, proteins, mRNA, metabolites, etc. You're breaking down in each of the individual components, and you're basically building up again the network, but in the form of a table. That was a table that I just showed you. I'm going to get back to the table. And each a member of that table is going to be one component of the network. And what you are doing is basically you are defining the interaction between the different components by these entries here. And in each case, by definition in this case, each of the components in the zeroth column is going to act upon the component in the zeroth row. So what we are saying is this complex here is negatively affecting the expression of that gene, and that's that's known. So you're going to have a limited number of, amount of information here. You're not going to know all the entries. You're going to know a very limited number of them only. However, due to the structure of this table, just the way we are building up the network, there are very nice relationships, and you can make inference about the entries that you don't have, about the cells that are empty. Meaning that you can start out with a basic or certain amount of knowledge about the network, and you can infer interactions from there on. And what is represented down here, we're not going to go too much in detail into it, but we did a lot of kind of training for this, basically showing that this thing can predict things that are known, let's say, without putting it into the table. And then here are a bunch of features of this tool, but I'm going to go down to the bottom of the list because it's the most promising feature or the most interesting. And the question is, can we use this tool for predicting something that we don't know about and that we cannot see from looking at the individual component of the network? And that's exactly what we did, and we did to investigate the role of MLC. This is a global regulator, but it's not known to be involved in the metabolism of cellulose, that is one of those 5-carbon sugars. And I'm going to blow up one portion of it. And the point is, we had calculated that it has a positive effect. Here is the global regulator. Here are the cellulose utilization genes. It has a positive effect on that. So it means that if we delete that gene, it should have a negative effect on cellulose utilization. And then that's what we did next. If this thing doesn't go back, I'm going to, okay, it goes. So we predicted that. Then we went and did the experiments. And that's exactly what we got. And let me show it to you in bigger scale here. One link is going to work. Let me go forward now. This thing always changes. Here is the experiment. What we predicted was that this is going to be required, this global regulator MLC is going to be required for cellulose utilization, and that's exactly what you get here. For the original cells, the one that don't have any modification, they metabolize both sugars, while however the mutant doesn't metabolize cellulose. The same thing happens when you put the two sugars together. So basically, by using that tool, that allow us to predict certain behavior, or I prefer to call emerging properties of that network, and we're able to predict the role of this global regulator that was not known at the time. So let's move to the other side of this diagram, and it's getting crowded, but again, the idea, and clicking every single part in here, so you actually don't need to see any of these things in here, just knowing that it's to the left or the right. Remember, I said that we use a lot of these omics tools to quantify level of expression of genes, protein, metabolic fluxes, and so on. And that's what we go and do down here. Now, I'm not going to continue with the part of sugar utilization, but I'm going to focus more now in the ethanol, the tolerance to ethanol of a given strain. That was also mentioned is a very important, it's a significant problem for the case of E. coli because it cannot withstand significant level of ethanol, so you get a very diluted product and so on. So what we did was basically we used DNA microarrays. We use DNA microarrays, and I'm going to skip the description of DNA microarrays. I'm going to tell you instead of the slide. These are the type of experiments we did. We have here 12 experiments, and we have a combination of different strains, 2 different strains that both produce ethanol, but one is more resistant to ethanol than the other one. Then you have different sugars, and then you have different level of ethanol in the medium. So you have a bunch of variables there. Then we did DNA microarrays for all those conditions. And when you do DNA microarray, what you are doing is identifying the expression level of each gene again in each of those conditions. And then the question was, out of all that data, we talk about more than 4,000 points for each of those conditions, how do you deal with the data? Well, here is where I want to show you another method that we have developed for that left, right of that cycle that I show you that allow you to infer, make inference based on the use of functional genomics tools, large data set, high dimensionality. The method is a PCA-based method. It's just got published now. And the idea is that you take all that data, what you have down here is the number of experiments, 12 of them, And then you are going, in a PCA-based method, you're going to have principal components out of your analysis. The first thing we do is we use this type of graph to associate principal components to the experiments, the strains, the condition in the environment, and so on. And then you can see, I'm just going to talk about, there are 3 principal components here, but let's talk only about the principal component 1. It's called, in this case, eigenacid, and we can talk more later on, but just for now, let's take it as a principal component. And then look at the diamonds. When you see the first 8 diamonds are aligning around -30, and the other 4 are up in 60. What this is telling you is that this principal component is differentiating between the first 8 conditions and the last 4. And then if you go back to the one that I showed you before, exactly the last 4 are the ones that were challenged with certain amount of ethanol. Then for the purpose of the presentation, I'm going to just give you an example of what we can do with that. The other components will represent something else. And the idea is you go and look into that component and see which is the weight that each of the genes, the contribution, I shouldn't say the weight because that's not what we want in here, in fact, the contribution of each gene to those components. And eventually, if that component represents response to ethanol, those genes that have a high contribution to that component are going to be related to the response to ethanol. And that's what we look at, and among other things, we implementing a method to define the genes that are contributing the most, that is based on the curvature of this curve here that is very steep. And basically, these are the response to presence and absence of ethanol, so 8 versus 4 cultures. Based on that, what we say is we identify the gene signature that is representing the response to ethanol. And analyzing what those genes are and all that, it makes a lot of sense that those responses were, in fact, some of the genes that we found in that way are related to metabolic responses that we had found before to be beneficial for ethanol tolerance. So that closes, I guess, this part of the cycle. And the message here is you use this type of approach, you use systemic approach, and definitely you go around this cycle and you come out with a better understanding of the system and updated knowledge. Now, next step is now you need to go and use that knowledge to improve utilization of sugar and to improve tolerance to ethanol. That, of course, I'm not going to get into, but we can talk about it later. Of course, it's not only about fuels. People have talked about biorefinery and producing chemicals and so on. I'm going to just show one slide about a project we have been working on for a couple of years. This is acetic acetate with several uses and all that. And I think that Richard was having in one of his slides acetic acid as one of the products. And what I'm trying to illustrate here is using metabolic engineering tools, we went up, and by now it's more than 90% the theoretical yield. This is different to what other people have done in the sense that it's anaerobic rather than aerobic fermentation that has high cost and so on. Now, I'm going to give you 5 more minutes, if I have it, in the oil platform. I don't work in the production of biodiesel. It is a chemical process, or mainly chemical, and then I don't do my research is mainly focused on fermentations and so on. The one thing I wanted to say here is that you have a common problem now that was a common benefit maybe a while ago, or maybe a year or 2 ago. That is that you have the byproduct in the production of biodiesel. You have glycerol as a byproduct, no matter how you go about it. You know that biodiesel is going up and up. I'm going to skip this one. You know that it's not a garage industry. If you don't know it, I want to tell you that it's not mix and match, and this very well established technology for it. Here to emphasize the point again that you get about 100 pounds of 95 to 100 pounds of biodiesel, you get 10 pounds of glycerin or glycerol. Roughly, you can approximate a little bit less than that, but it's a gallon and a pound. So 100 million gallon biodiesel in Galveston is going to have 100 million pounds sitting there. Well, hopefully not sitting there, but and saying sitting there because of this. Last year, about 40 cents per pound. Today, people are paying for disposing of this. Of course, people that are building biodiesel plants are really, really concerned about starting up their plant. They have enough problem with that. They're not thinking about this. Well, most people are not thinking about this, but it is a real problem. It's a real problem for them. It's a real problem for the people that make glycerin before, that are closing their plants. It's a real problem for the oleochemical industry that had this as a byproduct and made significant amount of money, and it's no market for this. Well, I'm going to give credit to the DOE. They put this report a year and a half or almost 2 years ago, and what they were predicting is about the oil platform, and they were envisioning that this was going to be a problem, and they were thinking that we were needing new glycerol platforms, but guess what? We don't have the new glycerol platform, but we do have the problem already. Why is glycerol? This slide summarizes a little bit of why it's important to find uses for this. Well, abundant, I told you already the numbers. Definitely it's going to be a renewable carbon source. It's highly reduced. I'm trying to put reasons here that are absolute. If the price of this goes up at the same price of sugar, it's still good to use this. Well, it's still good depending on the product you want. If you have a reduced product, and that's the case of fuels, right, where you need more electrons, these things has much more electrons than sugars. So definitely you prefer to use these. Well, the other ones I already mentioned, the price and, needed. I think that maybe I'm being a little bit bold here because I'm doing research on this, right? But I really think that this is going to have a huge impact in the biodiesel industry. People that were building biodiesel plants were thinking they were going to sell their glycerin, and now they need to pay for it. So definitely it's going to give some value. I think that we are in a pretty good position because we found that E. coli actually ferments glycerol, and the fermentation term here is strictly speaking in terms of the fermentation without electron sectors. And that's really important again, because if you want to take advantage of the highly reduced state of carbon in glycerol, you need to do fermentation. If you put an electron sector there, you're going to burn the electrons. It's going to go to your electron sector basically, and then you're not going to take advantage of it. But people thought for about maybe 80 years, I bet that I shouldn't bet anything, but most of you haven't gone to look at paper in 1925. I did it in Iowa. It was winter, it was really cold. It was in a building away from my office. So for sure, I surveyed the literature. What people think out there, what people thought, because we published it last month, was that this wasn't feasible, but it turned out to be feasible. There is a report. This graph to the left, intentionally, it doesn't have a legend. It's not that I miss it. It's just to show you that red line, those are the conditions that people used before we reported our conditions, meaning that the cells, this is the growth of the cells, and then if you put glycerol fermentation here, it's going to be flat. And then the other one is how we play around with it, and we got to a point in which it actually ferments glycerol. I mean, they grow, then they do ferment glycerol, definitely. We're not guessing that. Now, again, there's a great opportunity. Unlike the DOE platform, we don't have as many products as the DOE platform, but we still envision that this can be a platform and a new path to chemicals and fuels. And I have a bunch of products here. And in all the cases, doing this from glycerol is more advantageous than doing it from glucose based on the redox balance, because all of them are reduced products. And to the right there, I have the example of succinate. With very minor modifications, we are just increasing 5-fold the production of succinate already. One thing that you have, again, that it can easily illustrate the advantage of this over sugars is the production of ethanol from both substrates. If you produce ethanol from glycerol, you get ethanol and formic acid, or ethanol and hydrogen. From glucose, you will get ethanol and CO2. That's it, of course, because the oxidized state of the carbon on glucose is higher than glycerol. And then this is another example of basically the flexibility of these. So here you have fermentation that will be producing essentially, this is basically at theoretical yield, just in case you wonder which was the yield of these. This is basically hydrogen and ethanol. Doing a little bit of modifications, you can switch it, in fact, to doing ethanol and formic acid. I think that we're very excited about both ends, the tools that I presented before and the approach, that we're of course using all that approach in this type of project, and the potentials of these findings. And with that, I want to thank people that are doing a lot of work and supporting the research in our group. And I will be happy to talk to you later, maybe. You're not going to give me time for the questions, right?
Well, thanks. Thanks, Ramon, for a very nice presentation. Are there any questions for Ramon?
Is last process you talked about, this is a novel process? And if so, have you, just curious, have you applied for patents?
No, we both, the answer is both, yes. It's a novel process. Oh, sorry. It's a novel process. I mean, from the fundamental point of view, is a huge thing. That's what I think. And from the point of view of applications as well, and yes, we have pending patents.
And you think it's scalable?
It is. It is. Think about producing ethanol from that without breaking the corn. I mean, there's a limited feedstock of course, of glycerol. It's not for making a lot of things. If you make succinic acid, probably would make more money out of succinic acid than the money you make out of biodiesel in your plant. So. Thank you.
Thanks again. Let's move on to the next presentation, which is going to talk about something that has been around for a long time, actually since yesterday. Butanol production by microbial fermentation, and it's going to be presented by Professor George Bennett. And George is a professor and chair of the Department of Biochemistry and Cell Biology here at Rice, or is it Biomass University? George.
Okay, I was asked to give sort of an overview of the butanol situation or current status. So I'll try to give a sort of a wide ranging coverage of butanol. We've heard a lot about sustainability of these chemical processes, concerns about prices, environmental concerns. Quite a few studies have advocated these bio-based processes, particularly pointing out this European BioVision and the recent DOE roadmap advocating a significant contribution of biofuels. A recent announcement we heard about from Adam Schubert concerning DuPont and BP interested in the biobutanol formation. So he indicated a few of these advantages briefly, in that butanol has a lower vapor pressure, potential for pipeline transport, higher energy content, and able to blend in a variety of fuels over wide concentrations with low water absorption. In one sort of anecdote sort of study, this company indicated below drove an old standard vehicle around the country on essentially pure butanol without any engine changes. So let's look at the chemistry of these various alcohols compared to gasoline. Particularly, you note the vapor pressure of butanol is considerably less than ethanol and less than gasoline itself, while retaining a high octane number and energy calories per gallon, more comparable to that of gasoline. Another consideration of engines is this air-to-fuel ratio, which is also similar to that of gasoline. So it appears that it has some chemical potential for further study. Right now, the butanol market in the U.S. is not stupendously large, but butanol can feed into the production of other chemicals as well as being used as a fuel. So I'll go through some of the elements of this butanol story, going through some of the current organisms and pathways, a little bit of the history of the industrial technology, some areas of the feedstocks and current genetic and metabolic engineering routes to improvement. Many of these techniques were well covered by Ramon, and even more future-oriented techniques that are just coming into use. Some of the elements needed to improve this sort of production would be higher solvent concentrations. Many industries would rather work with a non-sporulating strain. Of course, the variety of products produced, you want a strain that produces almost entirely the desired product. In this case, that would be butanol. And it would be good if you could eventually integrate this with the use of cellulose. So while butanol production could be engineered into some other organisms, as far as I know, that hasn't been done. And I will cover some basic information about the organisms that do produce butanol. These are anaerobic organisms. Butanol is typically a major product, and quite a few strains have been isolated from all over the world which can carry this out. In a typical organism of this type, the organism usually produces acids early on in its culture, and then it will reach a point at which it begins producing primarily solvents and taking up these acids and converting them to the neutral compounds. This is thought to be a sort of defense mechanism against the buildup of acids, as well as a way to get rid of excess reductant energy. This sort of stage of the clostridial cell life, early on, you see the normal growth pattern, and then it goes into this sort of clostridial state where solvents are produced. And this is sort of correlated with the initiation of sporulation. In other sort of pathogenic Clostridium, this sort of sporulation state is often when toxins are produced. And these sort of processes of sporulation have been studied in a variety of organisms. As far as the enzymes and pathways for butanol production, this is summarized here through glycolysis. Here you then continue it beyond the 2-carbon state down to a 4-carbon activated intermediate, and over to either an acid or alcohol. And most of these genes and enzymes were cloned or analyzed here at Rice over the last couple of decades. So another attribute of these Clostridia are that many of these strains can utilize quite a few different feedstocks. There are strain optimization or strain preferences, but in many cases they can handle not only starches or sugars, but pentoses, materials present from a variety of waste food processing type materials as well. So one question is, can this sort of process be scaled up or taken seriously in an industrial context? So from the initial isolation of the Clostridium acetobutylicum by Chaim Weizmann, who then implemented it in the production of essential materials for explosive manufacture in World War I Britain, there have been quite a few instances where large-scale plants have been developed. I'll go through one example here showing this sort of vintage 1960 sort of technology in this relatively good-sized plant in Russia. This used a mixture of starch and also hydrolyzed biomass, produced about 4 million gallons of solvent annually. It was quite a well-integrated plant. They could use a variety of feedstocks here. They could collect not only the butanol, but also formulate some syngas, collect hydrogen as well, and some off use of vitamin production was also made. So this sort of illustrated here in some slides that Wolfgang Schwartz presented at the recent Clostridium meeting to sort of illustrate the relative quantities of materials. You also produce a lot of hydrogen in these sorts of processes. So this was a system which was able to do pretty large scale, also using some waste hydrolysates and some made use of the enzymes from the Clostridium for more effective digestion. So now let's look at the potential application of recent molecular biology or genetics to this process, as well as some of these engineering concepts Ramon discussed. So one thing is to sequence the genomes, find genes involved in the solvent production, analyze those gene expression protein levels, and then try to manipulate those in a way so that you increase the butanol proportion, rate of production, or the operating phase, or other useful parameters. Clostridial genomes have been sequenced. One of the first genomes that were involved in sort of the phase 1 of DOE sequencing areas here from the mid-90s was the R strain of the Clostridium acetobutylicum. More recently, another solvent-producing Clostridium has been sequenced, as well as some cellulolytic Clostridia and a number of pathogenic organisms from this genus. So once you have this sort of genomic information, you can carry out microarray experiments, illustrated here, one by our collaborator, Dr. Papadakis of Northwestern, which illustrates that the solvent-producing genes here correlate very closely with these very early sporulation genes, shown here in the red area, when they begin to be expressed in quantity. We've also carried out some protein analyses and observed differences in various strains and levels of enzyme or time courses to point out key enzymes in the process. So these sorts of studies allow some key genes to be identified, particularly a gene called SPO0A, various kinases, phosphatases, metabolic genes, which were known from early enzyme studies, and stress genes that also come on during high acid or high solvent production. So how can we modify these to improve our performance of the organism? One way in which that can be done is by constructing a plasmid that can replicate in Clostridium, and we've made a number of these with different selectable markers or origins. And then this can be recombined into the genome to eliminate activity of a particular gene. In this case, this was a regulatory mutant, and this produced considerably, maybe 30 to 50% higher solvent titers than the original organism. You can also carry out genetic alterations on metabolic pathways. These were some of our genetic engineering studies where we first showed that you could improve these characteristics through this sort of manipulation. Here's the butyrate kinase mutant. It shows a higher solvent production than the wild type here, and generally a little faster with lower proportion of other products. You can also overexpress some key enzymes from various plasmids, such as the alcohol dehydrogenases, using vector systems. And these can lead to increased solvent production or rates of production. One point concerning the ability of being able to prolong the solvent phase might be to decouple the tendency of the organism to sporulate from its solventogenic stage, since these are quite closely linked according to some regulation mechanisms, with some similar genes turning on both processes early on. What we wanted to do then was to block off sporulation at a relatively early stage. We focused on this phosphatase gene here. Thought if we eliminated this gene, we could decouple the two processes while retaining solventogenesis. Here compares the control strain with our modified non-sporulating strain here. This is under a non-optimal growth condition in a tube, but indicates that this sort of modification may allow larger operational windows and could prolong the solvent phase without bothering the ability to produce solvent. And this shows the control strain sporulating here, and most of these are not forming spores after a long period of time. Another point about production of butanol is how it can be integrated with other bioprocesses, other sort of industrial infrastructure, and how this can fit into larger ideas about long-term goals about using biomass for the ideal use. Ramon mentioned that biodiesel production generates a lot of glycerol. Here's a illustration of a use of glycerol by this Clostridial strain in production of butanol, published some time ago. But there's not been really a lot of support for work on a lot of Clostridial strains or this in general. So a lot of this has not really been carried forward that much. Here's some illustrations of the use of wheat straw hydrolysate recently by Nasib Kurshi, who presented at the recent Clostridia meeting hosted here at Rice in May, indicating that it produced these solvents in quite good character. Of course, the long-term ideal is to be able to connect these effective or the best production systems with cellulose. And I unfortunately missed the presentation by Professor Bayer this morning. I had to be at teach a class. But no doubt he covered the basis of the cellulosome occurring in a lot of Clostridial strains. This Clostridium thermocellum, these have been analyzed with quite a variety of enzymes that can degrade hemicellulase type materials and act on cellulose. So these are quite large and complicated systems. The genes for a number of these have been analyzed. Often there are big blocks of these together, which are kind of generally conserved among various strains. So this can allow potential for combining these useful attributes of cellulitic functions with some solvent production. The cellulosomes are pretty big, but they seem to be quite efficient at hydrolyzing cellulases. Another interesting industrially relevant point is with regard to product recovery, and I'm not really an engineer, but in work on gas stripping, this has led to increased term of productivity as well as higher sort of recovery of the solvent. So in summary, there are genetic routes to improved performance of these sorts of organisms beyond those used in earlier times, which were naturally isolated strains. There seems some potential for integrating this into other sort of bioprocessing infrastructure using hydrolysates of various sorts, or potentially relying more on the endogenous hydrolases of Clostridia. And the more we find out about these sort of organisms, the more we can do more with them. I'd like to mention the workers on the area, our longtime collaborator, and those people who presented at the conference and gave me extra information. Thank you.
Well, thank you, George, for a very nice presentation. Are there any questions for Professor Bennett? Well, I realize we have run way over schedule, and I apologize for that. But we have built a 1 hour general scientific discussion in our afternoon program, so we can probably pick up some of the Q&A then.